In 1665, Robert Hooke, a natural philosopher, designed and constructed the first light compound microscope, consisting of two converging lens systems, the objective and eyepiece. He used this microscope to observe cork, and as he saw the building blocks that made up the dead plant material, he discovered and named the cell. Around 1670, Antoine van Leeuwenhoek constructed a single-lens microscope by crafting very thin glass lenses so that they could magnify by hundreds of times. The lens is placed onto a metal plate and used transmitted light to create an image in the drop of water at the end of a metal pin, which was moved back and forth in order to focus the image. Through this microscope, he was the first to observe and describe living cells. Up until this point, only light microscopes were used, but in 1931, German physicists Max Knoll and Ernst Ruska invented the electron microscope. While a light microscope uses a beam of light to illuminate and glass lenses to magnify, an electron microscope uses a beam of electrons to illuminate and a magnetic lens to magnify, resulting in greater magnification, magnifying up to 1 million times, and a greater resolution. One drawback compared to the light microscope is that the radiation from an electron microscope destroys living organisms, so today we will still use the light microscope to observe living cells.
When talking about electronic microscopes, there are two different types of them. There is the SEM and then there is the TEM. The TEM is a transmission electron microscope which fires a beam of electrons through a specimen, while the SEM, the scanning electron microscope, scans a beam of electrons over a specimen to produce a magnified image. Now how they both work is that a stream of high voltage electrons, which is formed by an electron source, which is usually heated with a tungsten filament, is accelerated in a vacuum using a positive charge. This stream is confined and focused using metal apertures and magnetic lenses to a thin focused beam. This beam is focused onto the sample using a magnetic lens, and interactions inside the sample affects the electron beam. These interactions are detected and transformed into an image which is processed so we can see.
An application of an electron microscope are for viewing structures and composition of everyday material that we encounter and get to view it in a nanoscale. To put a nanometer in perspective, here is a ruler. Right here we have 1 centimeter. From 1 centimeter there are 10 millimeters, and from one of those millimeters there is 10 micrometers. From one of those micrometers are 10 nanometers, and there is our scale for how close we can get with an electron microscope. Here is an example of a gold on a sample plate. To the naked eye, it looks like a sprinkle of gold dust. However, under an electron microscope, we clearly see the surfaces and structures of our gold in different shapes. We can further our understanding of compositions of elements and biological matter, from viruses to butterfly wings, all under the electron microscope.